Okay, so we left off with this slide. I'm going to try to make this lecture a little bit shorter. Please keep in mind that, again, all of the information that I'm putting in these lectures, I put a lot of information in there because you guys don't have textbooks. But please keep in mind that I'm going to test you at the level at which I'm going to talk about this in my lecture. Okay, so we left off with this slide here. We're going to talk about the difference between action potentials in neurons versus muscle cells. And remember that when we talked about muscles, we talked about action potentials, again, moving across that sarcolemma, right? And so, but we didn't really talk about what the action potentials were because I knew we were going to get into it here. So now, this is just to remind you that action potentials, again, happen across muscle cells as well. And it's going to happen across the entire muscle cell. Instead of, in a neuron, remember action potentials are only happening in the axon, right? So that's where we said action potential axon, right? Both start with A's to help you remember. And so there's a couple other differences in the action potentials in neurons versus muscles. One is the resting membrane potential of a neuron is, again, minus 70 millivolts. We know that. But in skeletal and cardiac muscle cells, it's actually closer to negative 90 millivolts. Okay? So again, a lower resting membrane potential. Also, you probably intuitively know that nerve impulses, again, are going to be faster. So the duration is going to be shorter for a nerve impulse. A nerve impulse is about half to 2 milliseconds, okay? Versus the muscle action potential will last anywhere from 1 to 5 milliseconds. For if you're talking about skeletal muscle cells, and then anywhere from 10 to 300 milliseconds for cardiac and smooth muscle cells. So again, like a much larger difference. So in terms of the duration, again, nerve impulses are going to be the shortest. They're going to be the fastest, right? And then skeletal muscles are next, and then cardiac and smooth muscles are last, okay? So the fastest nerve conduction is actually 18 times faster than that of a skeletal muscle fiber. Okay, so again, and it makes sense, right? Nerve conduction should be faster, right? We need to transmit that information really rapidly. So now let's talk about signal transmission at our synapses. So remember that a synapse, again, is that junction between one neuron and another neuron, okay? Or a neuron and its effector, which would be a muscle cell, or a neuron and its effector, a gland, okay? So that's your synapse. And we've already talked about a synapse because we talked about the synapse between a neuron and a muscle cell already. So this actually should be pretty easy, and I'm going to go through this pretty quickly. There are two types of synapses. There's an electrical one and a chemical one. The electrical one, you can kind of picture this is going to be the common synapse that you're going to see in like cardiac muscle cells. And this is where an ionic current is going to be spread from one cell to the next cell through gap junctions. So these cells are going to be connected just like you see in your cardiac muscle cells, right? They're going to be connected at that intercalated disc, and they have gap junctions. And so again, as sodium's brought in, it actually will move from one cell in the interior of one cell to the interior of the next cell through these gap junctions, okay? So this is a really fast way to transmit, again, that electrical impulse. And it's really capable of synchronizing groups of neurons or, again, groups of, cardiac muscle cells, okay? And then a chemical synapse, which is, again, kind of the more common one that we talk about, is where we have this kind of one-way information transfer from what we call a presynaptic neuron, right? That's the neuron before the synapse, right? And then the postsynaptic neuron, the neuron that it's going to be kind of passing its information onto, okay? If you can picture that. So that's the two types of synapses. And then I'm going to go through this in just a minute, and we'll talk about the nitty-gritties on how things happen, how signal transmission happens at the synapses. So I'm going to kind of come back to that. But really quick, before we get into that, I wanted to remind you of the difference between graded and action potentials. And on the last lecture, I didn't have like a really organized slide on this. So I put this together so that way then you have this nice organized slide on the differences between graded and action potentials. So remember that graded potentials are going to arise on dendrites and cell bodies. And action potentials are going to arise only at the axon hillock. Okay, so that's one of our major differences. Two, the types of channels. So an action potential, again, is going to be produced by opening these voltage gated ion channels, right? Because we hit that threshold of like negative 55 millivolts with that, again, a voltage change that occurred. And so action potential, again, requires our voltage gated ion channels to open. Now, in our graded potential, remember that happens in the dendrites and the cell body. Remember that this is going to be produced by a ligand or chemical, again, gated channel. So something like a neurotransmitter binding, again, to a gated channel and opening that channel, or to a mechanically gated channel. Okay, so again, maybe you put some pressure and you kind of deformed that gated channel, and that allowed, again, the sodium to rush in. Or it could be potassium rushing out if it's inhibitory, right? So that's another main difference. 3, conduction. Graded potentials, right, are very localized. They're not propagated down an axon. Again, they're very localized to that one area. Action potentials, of course, excuse me, are going to be just the opposite, right? An action potential is that all or none. And so, again, once an action potential is initiated, it's going to conduct over the entire surface of the axon. There's no stopping it, right? It's like a freight train. Number 4, amplitude. So the amplitude of the action potential is always going to be constant, right? Because it's all or none. So again, either you're going to, go all the way for that action potential, or again, not at all. The graded potentials are going to vary depending upon the stimulus. And so again, this is how our graded potentials can eventually lead to an action potential. We're going to talk a little bit more about this in this lecture. The duration of the graded potential is as long as the stimulus. So this is your 5th one, duration. The duration of the graded potential is going to be as long as the stimulus lasts, right? So this is going to be different for your action potentials, because your action potentials have a refractory period. So again, you have the action potential, but then you have this time period, right? Where even if the stimulus is still coming through, that neuron is not going to, that axon is not going to be able to generate an action potential for this short time, that refractory period. And so that's the 6th one, which is that, again, an action potential has a refractory period and graded potentials don't. So graded potentials can keep getting the stimulus, right? And it'll keep causing those gated ion channels to open, versus, again, an action potential has this refractory period where, remember, those sodium inactivation gates are closed. And so there's a short time where they can't be opened. So I liked this this meme here, as we're not the same. So again, graded potentials and action potentials are very different, right? And this kind of sums up, I think, everything. And I'm going to come back to this a little bit later in this lecture just to show you this. But I love that it sums up that here your graded potentials, right, are going to be happening in the dendrite and in the cell body here. And what's interesting is actually your excitatory potentials, right, that are going to cause depolarizing graded potentials, I should say. Those actually are going to happen with a presynaptic neuron that's synapsing with the dendrites here. Okay, so those are going to be the excitatory ones. Now, inhibitory ones are usually coming from neurons, presynaptic neurons that are synapsing on the cell body. So just kind of interesting fact for you there. But again, remember, we're going to talk about this process, this idea of summation, where basically summation, the addition of, right? The addition of all of the excitatory, graded potentials, and then the inhibitory graded potentials are going to be kind of add it up all together. And as that happens, I love this. You have like some excitatory ones, right? That are it's like, so some of these ones here are firing and causing some depolarization, but then you got some inhibitory ones that kind of take you back down a little bit. But then you got more of these excitatory ones and they're firing, and then maybe you got one that takes you back down. But then you've got, again, because you have more of these excitatory graded potentials and excitatory presynaptic neurons, then eventually they'll get you to that negative 55 millivolts. And again, once we create enough of a change to where we hit negative 55 millivolts here in that threshold, boom, it's action potential, all or none. We're, again, going to cause that again, signal to be sent all the way down the axon. Okay, so again, I really like this diagram. I think it really helps to kind of put it all together. Now, let's talk about what happens here, okay, between this presynaptic neuron. And remember, again, that presynaptic neuron is going to end in the synaptic end bulbs. So what happens between the synaptic end bulb of our presynaptic neuron and the dendrite here of our postsynaptic neuron? And it's actually pretty easy. Again, especially because we've been through this really with, again, that neuromuscular junction, right? So just like before, we're going to have a synaptic cleft here, but this time it's between, again, this synaptic end bulb of a presynaptic neuron, and here this is going to be your postsynaptic neuron membrane. So no motor end plate here because it's, again, this is just another neuron. But this postsynaptic neuron in the dendrites, or again, the cell body, is going to have these gated ion channels. And in this case, again, it's going to be these ligand gated channels if it's going to be, again, a synapse between two neurons. So what happens is as that nerve impulse travels down the axon and then travels into these axon terminals and then into the synaptic end bulb, what's going to happen is you're going to have these voltage-gated calcium channels at the end. So as voltage-gated sodium channels are opening here and allowing sodium in, that's causing, again, a change in the resting membrane potential here. Then it's going to trigger these voltage-gated calcium channels now to open in the synaptic end bulb. And as that calcium rushes in, that is going to stimulate the release of neurotransmitters. So it's calcium coming in here into the synaptic end bulb that stimulates the release of neurotransmitter. As neurotransmitter is released, it's going to diffuse down its concentration gradient across this synaptic cleft. Remember, that's the space between the two. And then eventually bind to this postsynaptic neuron, right? And these, again, ligand-gated channels. And as it binds neurotransmitter, it's going to cause these ligand-gated channels to open, allowing something like sodium to rush in, okay? And as that happens, again, now you're causing here an excitatory, if it's sodium, right? It would be an excitatory graded potential here in your postsynaptic neuron. So again, these are going to be our chemical synapses, okay? And so just like I talked about here, again, the action potential reaches the synaptic end bulb. As it does, it's going to stimulate these voltage-gated calcium channels to open. Calcium, again, as it flows in, is going to trigger neurotransmitter to be released across this synaptic cleft. And then that neurotransmitter, again, will bind to these ligand-gated receptors on these chemical-gated channels, allowing those chemical-gated channels to open and allowing ions through. Okay, and then, again, ultimately, it can potentially lead to depolarization in that next neuron. Okay, but again, this is gonna be causing that graded potential, right? Because here, your synapse is going to be between that synaptic end bulb and either, again, a dendrite or the cell body. So you're either, again, causing an excitatory or an inhibitory graded potential here. Okay, so really quick before we leave this synapse, I want to talk about what happens with neurotransmitter here. So we do have to remove neurotransmitter, right? I mean, we can't just keep neurotransmitter here. We just continue to keep firing this next neuron. And that would be bad, right? Because we want neurons to fire sometimes, and sometimes we don't want them to. So we have to, again, have removal of the neurotransmitter here. So what happens is there's three ways that like neurotransmitter ends up getting removed. And it's a combination of all of them that are responsible for the removal of neurotransmitter. So one is simply just diffusion, like the neurotransmitter in this synaptic cleft is eventually just going to kind of diffuse out, this way and this way here down this concentration gradient. Also, enzymatic degradation. So we saw this already with neuromuscular junctions where we had enzymes out here that are going to degrade the neurotransmitter. So for example, acetylcholinesterase, remember, was released to be able to degrade the acetylcholine neurotransmitter at a neuromuscular junction. So we'll have, again, other enzymes that will degrade other neurotransmitters. And then the third one, and this is kind of important for understanding how certain drugs work, is that the reuptake of that neurotransmitter by either the presynaptic neuron itself. So sometimes this presynaptic neuron will actually take back up some of the neurotransmitter that's left over here in that synaptic cleft, or sometimes the glial cells, especially like astrocytes, will come and take back up that neurotransmitter so that it's not just hanging out there in the cleft. So there are certain drugs that actually work to prevent the reuptake of that neurotransmitter, letting the neurotransmitter like hang out in that synaptic cleft longer, allowing again for that postsynaptic neuron then to be stimulated, more frequently. So, for example, Prozac is a serotonin reuptake inhibitor, and it prevents the reuptake of that serotonin neurotransmitter and lets it hang out in that synaptic cleft longer and be more available again to bind and continue to cause change. Again, oftentimes depolarization in that postsynaptic neuron. Okay, so those are just the ways that we get rid of neurotransmitter. Now, again, we've talked so much about this, but again, I just put this in another slide here for you, so you've got it. The effect of a neurotransmitter can be excitatory or inhibitory. And believe it or not, the neurotransmitters that are inhibitory are just as important as some of the ones that are excitatory. And you're going to see this, excuse me, when we talk about neurotransmitters in just a minute. Excuse me for a second. Sorry, I'm starting to lose my voice there. Again, an excitatory, what we call an EPSP, is an excitatory post-synaptic potential. So that's what EPSP is. And then IPSP would be inhibitory post-synaptic potential, okay? So an excitatory postsynaptic potential is obviously going to be one that is going to cause a depolarizing effect, okay? So in this case, again, it's going to bring us closer to thresholds, okay? And so again, of course, these are going to be graded potentials here. This is graded potentials because again, axon potential only goes one way, right? So again, a depolarizing postsynaptic potential will cause these ligand-gated sodium channels to open, allowing sodium to rush in, and again, bringing that interior of the cell closer to that threshold point, okay? Then an inhibitory postsynaptic potential is going to do just the opposite here. So just like this here. Instead, and remember that the excitatory ones are oftentimes, again, synapsing with dendrites. Here, the inhibitory ones are oftentimes synapsing with a cell body. Why? I don't know, but it's just how it is. And So the inhibitory ones, because they're bringing us, again, farther away from threshold, they're making it more negative in the interior. So they're going to do things like open ligand-gated chlorine channels. That's a negatively charged ion to allow negatively charged ions in, making it more negative. Or potassium is going to open a potassium-gated channel, allowing potassium out and making it, again, more negative on the inside. Okay, so then in these inhibitory postsynaptic potentials, these IPSPs, the postsynaptic cell is going to be less likely to reach threshold, right? Because we move farther away from threshold. Okay. So, and we already kind of talked about this. I'm going to go through this quickly, but summation then is just basically, the addition of all of the graded potentials together. So all of your inhibitory and your excitatory potentials together. And is that, again, when you add all these up together, is it enough to hit threshold or is it not? And so that's what summation is. Now, summation can be either spatial or temporal. Oh, and so really quick, I blew this up for you because I think this is such a great image of summation. So again, if you have more excitatory postsynaptic potentials than you do inhibitory postsynaptic potentials, right? Then that will allow you to have enough of a change that, again, at that axon hillock, at the trigger zone, you can actually reach that threshold of negative 55 millivolts, okay? So that an action potential can be triggered all the way down that axon. So again, this is just a great image of showing you summation. Now, summation can either be spatial or temporal. So spatial is just kind of the summation of the effects of, again, multiple different synaptic end bulbs that are contacting this, presynaptic. I'm so sorry, I said postsynaptic. It's going to be the summation of all of these presynaptic end bulbs onto this one neuron. Okay, so it's spatial because, again, there are many of these different neurons. And so how many of these neurons are excitatory and how many of them are inhibitory that are actually, again, spatially contacting this postsynaptic neuron is going to make a difference on whether or not you can meet threshold, right? And which of those ones are actually active as well. So again, maybe you've got, one excitatory synaptic end bulb here, but it's not enough to bring you to threshold. But then you got another one over here, and then another one over here, and another one over here. And that's enough to cause, again, enough of a change in that resting membrane potential to hit threshold. Okay? So that's all that spatial summation is. So again, another instructor gives this example, and I actually love it. So let's say that, you have, with social media, because everybody uses social media today, right? Let's say that you have like 100 friends that are telling you, yes, go do something, and only 2 friends, tell you not to do it. They're the inhibitory ones. Then chances are you're likely going to go do the thing, whatever it was that, all your friends want you to do. Okay. Now, temporal summation is, again, where you have the sum of firing over time. Okay, so temporal is over time. And so this is where we're talking about, again, the effects of a neurotransmitter being released from the same end bulb over and over and over again in rapid succession. Okay. And so again, if it's continually firing over and over and over again, and remember in your graded potentials, that can happen, right? Because there's no refractory period. So again, what will end up happening is, you bring a little more sodium in, it keeps firing more sodium, more, until eventually you hit threshold, okay, just from that one neuron firing so rapidly in rapid succession. So again, this is kind of like, if you've got a spouse kind of nagging you, take out the trash, 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 and they keep going over and over and over. You eventually like, fine, I'll take out the trash, okay? And then boom, you hit action potential. So that's kind of a good example of that. Okay, so again, you have both excitatory and inhibitory neurotransmitters in both the central nervous system as well as the peripheral nervous system. And the really crazy complex part of this where it just gets wacky in terms of trying to keep up with what's going on in the nervous system is that this very same neurotransmitter can be excitatory in one location, and then it binds to a receptor in a different location, and that receptor is inhibitory. Okay, so that's the crazy part. So these neurotransmitters can be excitatory in some areas of the body and then inhibitory in others, right? Totally crazy. But that's how it works, and it's very well controlled. So we're going to talk about some of these neurotransmitters that are really important, things like acetylcholine, glutamate, GABA, which is gamma-aminobutyric acid, glycine, norepinephrine, epinephrine, and dopamine, because these are all important neurotransmitters. And then really quick, there are 4 ways that neurotransmitters can be modified. So one, our body can influence the actual amount that's synthesized. So the synthesis can be stimulated, so you produce lots of that neurotransmitter, or, it could be inhibited if it needs to be. Also, the release of that neurotransmitter can either be blocked, or that release of that neurotransmitter can be enhanced, where you can, again, it allows you to release lots of neurotransmitter. The removal of that neurotransmitter from the synaptic cleft, again, can be stimulated or blocked. Again, we can do this with things like reuptake inhibitors, right? That block the removal of that neurotransmitter and leave it hanging out there in the synaptic cleft. And then the receptor site itself can actually be blocked. Sometimes, again, a lot of drugs do this. We'll have certain drugs that will bind to and block that receptor site so the neurotransmitter can't bind to it. Or again, they can activate it as well. So some drugs will actually bind to it and activate it. So again, the reason that these neurotransmitter effects are important is because there are oftentimes ways in which we can modify the effects of neurotransmitters, especially with certain medications. And just like before with muscles, we're going to use the term agonist for anything that enhances the neurotransmitters effect, just like, again, we did with muscles enhancing. And then antagonist is anything that's doing the opposite or blocking the action of the neurotransmitter. So again, just the same terms, just kind of reminding you. So let's get into some of these neurotransmitters. So thankfully, we've already done acetylcholine. Acetylcholine, again, is released at that neuromuscular junction. And again, it's excitatory at the neuromuscular junction, but other areas in the body, it could be inhibitory. Okay, so kind of interesting, and you'll kind of we'll get into that a little later. But again, you should know acetylcholine for sure for this next test. It's excitatory at the neuromuscular junction, right? So again, muscle binds acetylcholine, it's going to be excitatory. Some amino acid neurotransmitters. So glutamate is a really big one. And glutamate is your big excitatory neuron in the brain. So it's a really, really important excitatory neurotransmitter. I think I said neuron. But glutamate is a really, really important excitatory neurotransmitter in the brain. Okay? So you should know that one. Glutamate, excitatory in the brain. GABA, okay, is going to be the main inhibitory neurotransmitter in the brain. And so this is really, really important for, again, inhibiting too much neurotransmission. So again, if you didn't have any GABA neurotransmitter, you oftentimes, or if you're not producing enough GABA, you can end up having seizures and things like that. Okay, so GABA is the main inhibitory neurotransmitter, and it's inhibitory for about 1/3 of all brain synapses. So again, it's important because we don't want neurons firing all the time. There are times where we don't want them to fire, and so we need these inhibitory neurotransmitters. Just in terms of some drugs, interesting, Valium is actually a GABA agonist, so it actually again enhances the effects of the GABA neurotransmitter. And so by enhancing, again, that activity of GABA, and things like Valium as well as Xanax, it can do things like decrease muscle tremors, decrease anxiety, and things like that. So again, kind of interestingly enough, I'll just share with you, I have a, a, a condition, an interesting kind of condition called Meniere's disease. And sometimes, again, unfortunately, those neurons that are bringing information from the inner ear become too excited. There's too excitatory, and it'll actually cause, I'm getting too much information kind of coming back and forth across those neurons and no inhibition, and it actually will cause really bad vertigo spells. So sometimes one of the solutions is to take, mild doses of Valium to kind of inhibit the transmission on some of those neurons. So just kind of interesting information. And always interesting when you can kind of apply this to, start to apply this to things that, your parents are taking or things like that. It's just really interesting. There are lots of these biogenic amine neurotransmitters. And so one example of one, these just like, again, these modified amino acids. So one is norepinephrine. You've probably heard norepinephrine or noradrenaline. This is going to be really important for, again, regulating your mood, really important for like kind of awakening you, arousing you, waking you up from deep sleep, and things like, again, dreaming. So again, lots of different drugs that we're going to talk about. The release of norepinephrine is actually enhanced by amphetamines. And then another drug that kind of, again, is going to enhance the effects of norepinephrine as a neurotransmitter is going to be cocaine. Will actually block the removal of norepinephrine from that synapse, okay? So again, it's going to allow that norepinephrine to hang out there even longer. So amphetamines and things like cocaine are actually going to enhance the effect of that norepinephrine neurotransmitter. Dopamine, again, is another mood enhancing neurotransmitter. It's your feel-good neurotransmitter, right? And then we also don't talk about it very much, but it actually helps to regulate skeletal muscle tone as well. And so in things like Parkinson's disease, what ends up happening is kind of a neurodegenerative condition with where you're actually deficient in in dopamine and these dopamine receptors. And what ends up happening is you again start to see some of those issues with skeletal muscle tone. People end up having tremors and aren't able to maintain their skeletal muscle tone. And so a lot of times they'll actually give these patients something called L-Dopa, which enhances, again, the effects of that dopamine, enhances the release, actually, of that dopamine from the presynaptic neuron. So, again, L-Dopa and amphetamines will enhance the release of dopamine. Also, cocaine, again, just like before, is going to block the reuptake of dopamine. So again, cocaine does that for both of these, blocks the reuptake of norepinephrine, blocks the reuptake of dopamine, and then you're going to see amphetamines are going to work on all three of these, okay? So again, amphetamines are going to enhance norepinephrine, amphetamines are going to enhance the release of dopamine, and then, before I leave dopamine behind, kind of interestingly, they've found out that dopamine neurotransmission increases in cases of schizophrenia. They don't truly understand all of that and why it happens, but just kind of interesting fact for you. And then serotonin, again, and these neurotransmitters, I should mention, do a lot more. We're just kind of giving you the very broad responses for these neurotransmitters. There are actually other things that these neurotransmitters will stimulate, but these are just kind of the biggies. Serotonin, again, another mood controller. So you can see all three of these are associated with mood. And then serotonin actually impacts your temperature regulation, in induction of sleep. So again, it helps you to actually fall asleep. And then it also helps control appetite. So again, serotonin is, of course, the release is going to be enhanced by amphetamines. So amphetamines enhance the release of all three of these. And then serotonin reuptake inhibitors, like we talked about, are again going to prevent the reuptake of serotonin and kind of let it hang out there in the synaptic cleft a little bit longer. And again, that oftentimes helps to relieve anxiety and depression in individuals that aren't naturally producing enough serotonin. So again, in oftentimes cases of like clinical depression, for some reason that individual, and there are lots of different causes as to why, but for some reason that individual isn't producing enough of their own serotonin. So what they do is they'll give you these SSRIs, which are these serotonin, basically the serotonin reuptake inhibitors, and that allows that serotonin to hang out in that synaptic cleft longer since you're not producing enough of your own. And then, it basically allows you to kind of act at a more normal level of what would be serotonin with these serotonin reuptake inhibitors. And again, that really helps to relieve anxiety and depression for some of those mood disorders. Some other neurotransmitters are things like ATP and purines. Some of the other purines are going to be, again, this is adenosine triphosphate, adenosine diphosphate, adenosine monophosphate, and adenosine. So all of these, just all you have to know for now is that they can be excitatory or inhibitory. Now, I realized in your lecture, for some reason, it just says excitatory. So you want to go ahead and add this into your lecture. They can be excitatory or inhibitory in both the central nervous system and peripheral nervous system. So it just depends on where they're at. And they're oftentimes released with other neurotransmitters. So it's something like acetylcholine or norepinephrine. So again, you just kind of have to know these at the really basic level that they can be excitatory or inhibitory. They're in the CNS and the PNS. And then this I just thought was interesting because I love coffee. So adenosine is actually a really potent inhibitor in the brain. And so caffeine actually goes and it blocks these adenosine receptors. Hence why it's such an awesome stimulant, right? So there you go. Have your cup of coffee. And then gases. So we're not going to talk about these a lot right now, but you will in 202. And especially when you get to the cardiovascular system, because something like nitric oxide actually acts as a vasodilator, a really important vasodilator that helps to kind of lower blood pressure when we need to. So when we need our blood pressure to lower, nitric oxide will be released as this neurotransmitter in the form of a gas. We've also found that some of these gases are important for memory and learning. So kind of interesting as well. And then neuropeptides are going to be these neurotransmitters. They're still neurotransmitters. So again, neuropeptides are just another category of neurotransmitters that consist of about 3 to 40 amino acids that are all linked together again to make these peptides, hence the name neuropeptide. And some examples of this are like substance P. And so this one's easy to remember. Substance P, think P, pain. This is going to enhance our perception of pain. That's a neurotransmitter that's going to enhance our perception of pain. So as you can imagine, our body targets substance P on its own for pain relief by producing some of these natural pain relieving neurotransmitters. So enkephalins and endorphins are both going to be these pain relieving neuropeptides that are going to actually block the release of substance P. So kind of cool. Again, we produce these naturally. So again, that's why we call these our natural opiates. Our enkephalins and endorphins are natural opiates. And the effects of these two molecules are usually, again, mimicked by our drugs that we take. So things like morphine and heroin and methadone. And opiates and morphine are a little bit different than your natural enkephalins and endorphins because they actually are going to bind to that receptor, but they end up producing a stronger effect than these two do in terms of, again, blocking that substance P. So again, you guys probably all have experienced endorphins at some point in time. This is known for what's called your runner high afterwards. And so, if you have ever, I like to run half marathons and things like that. And so you're like running the half marathon, and at the end of it, you're like, my muscles aren't feeling all that bad. And I'm at the end of the race. Yeah, way to go. And so you finish the race and you're like, my muscles are tired, but they're not hurting at all. I'm not feeling that much pain. And then as the endorphins start to wear off, you're like, whoa, yeah, now I feel it, right? Or you may have done this if you've ever done, some activity that's really intense. So, maybe you're climbing or something like that, and you fall and you or really, bang your leg up, and you're like, oh, I don't really feel that. I'm just going to keep going. I'm fine. And then all of a sudden afterwards, you're like, whoa, now I really feel it. And if you think about this is kind of our body's own natural way over time of developing, and over evolution, I should say. Evolution is really kind of selected for this because over time, when we were back in the days of hunter-gatherer and, you're out there and all of a sudden, the scary bear is coming to chase you down, right? As you're running away, you don't want to if you fall and, hurt your ankle, you don't want to have pain prevent you from running away. So your body releases these endorphins and it's like, oh, you're fine. Keep running. Go, go, go. And that way then you don't feel the pain as you're running away from the scary bear. So again, kind of interesting. And so again, of course, because we have these natural ones, actually the term endorphin, this is really interesting, actually means endogenous, basically your endogenous morphine. So it's naturally produced, endogenous, meaning produced on its own in your body. So it's interesting that, again, we just use these natural pain relieving neurotransmitters and then mimic the effects with these artificial ones when we need to. Okay. Now the challenge, of course, with these is chronic use. So what ends up happening is if you are chronically using these, again, artificial pain relieving neurotransmitters, what ends up happening is your body's like, oh, well, I have enough of that. I don't really need to produce these endorphins on my own. I don't really need to produce these enkephalins on my own. So again, that can be some of the challenges with, again, chronic use. So it's really like a challenging kind of balancing act with those. Now, acupuncture can actually produce loss of pain sensation because of the release of actually opioid-like substances such as endorphins from acupuncture. So there is some actual science behind the use of acupuncture for pain relief. Really interesting. Okay, other neuropeptides, cholecystokinin. Cholecystokinin is a good one to know because it's known as the gut-brain peptides. They're found in the brain and in the small intestine. These are the neurotransmitters that are responsible for our stop eating signal, like, okay, I'm full, stop eating. And they also regulate enzyme secretion by the pancreas, as you'll get into this more in the digestive system. The contraction of the gallbladder to release, again, your bile that's going to help break down fats. And so again, I like to introduce that because you will talk about cholecystokinin, CCK, in 202. So, and then the last one I'm going to talk about is the endocannabinoids. They act at the same receptors as THC, again, which is the active ingredient in marijuana. And they're lipid soluble. They are, again, synthesized on demand, and they're believed to be involved. We don't really truly understand all of what they're involved in yet, but we have some pretty good data to show that it's involved in learning and memory, also involved in neuronal development, helping to, again, control appetite, and even helping to suppress nausea. So kind of interesting. Now, remember that these neurotransmitters are going to be, some of them in some areas of the body will be excitatory and then inhibitory in other areas. So again, kind of interesting. GABA and glycine, which we didn't really talk about, but these are almost always inhibitory. Okay, so those neurotransmitters are inhibitory. Glutamate, we talked about excitatory, right? That was the excitatory one in the brain. Got to know glutamate, excitatory in the brain. Acetylcholine and norepinephrines, those ones just depends on where they're at in the body. So for example, acetylcholine is excitatory at neuromuscular junctions for skeletal muscles, but it's actually inhibitory in cardiac muscles. Crazy, right? So again, the effects of these neurotransmitters just depends on where you're at in the body. Now, some neurotransmitter disorders. So, for example, in things like ADHD, some of the drugs that they have, most of the drugs that they have are actually impacting things like your norepinephrine and your dopamine levels. And so for ADHD, all of these drugs are going to, in some way or form, enhance the effects of norepinephrine and dopamine. So, for example, Ritalin actually increases norepinephrine and dopamine levels by reuptake inhibition. Kind of interesting. And I'm not going to test you on these. I put this on here because it's interesting. Dexedrine is going to act like an amphetamine, and it's going to increase norepinephrine and dopamine as well as serotonin levels. Adderall is going to do the same thing, increasing all three. And then Strattera is going to be this kind of selective norepinephrine reuptake inhibitor, just basically only, again, preventing the reuptake of norepinephrine. So again, kind of interesting. Depression, Cymbalta is a serotonin norepinephrine reuptake inhibitor, same with Effexor. Okay, so just kind of interesting in terms of some of these different disorders. Now, we don't truly understand all of the initial the drivers, I should say, of depression. So there are lots of other theories out there in terms of what are some of these initial drivers of depression, and those are things that are being studied in terms of new medications. So we already talked about this kind of net summation. So I'm not going to talk about this again. We already kind of covered that. I'm also not going to cover neuronal circuits just because we have really had so much in this lecture, and it's just not going to get us a lot of information for what we need right now. But I put it in there if you want to look at it. I do really quickly want to talk about regeneration and repair. So we do know that there's this thing called neuroplasticity that's maintained throughout our life in the central nervous system and peripheral nervous system. And that is that our existing neurons can actually sprout new dendrites, and they can actually change their synaptic contacts with the neurons. So in especially the central nervous system, we'll talk about the ability to produce new neurons is very, very, very limited. However, we can make new connections with our existing neurons, and that's what's cool. And so this is where they're talking about, again, that neuroplasticity of making these new connections between neurons. And obviously, as you use, if you're constantly using a certain connection between neurons, then that connection is going to get stronger. It's going to sprout more dendrites, right? And make more contact with that neuron. And so that's kind of that idea of like use it or lose it. So that's why they say, as you get older and you retire, they're like, continue to do things that are going to challenge your brain because of this idea of neuroplasticity, you'll maintain these connections and make these connections stronger if you're continuing to use these connections that are causing, higher cognitive functions in your brain. So that's that idea of, if you don't use it, you'll lose it. That's where that comes from. Now, when we talk about regeneration, repair, remember that the peripheral nervous system could do this. We already talked about that a little bit with those Schwann cells that are wrapped around the axon. So if there is damage, especially to the axons, that can be repaired in the peripheral nervous system, where the central nervous system doesn't have a repair system in place for that. And so regeneration is very limited. Also in the central nervous system, until recently, we just thought that their neurons couldn't, regenerate, so we couldn't produce more neurons. There are very few places in the brain that you can do this, but there are a few places in the brain, and I'll talk about that, okay? So again, repair within the peripheral nervous system is much more common because we have these Schwann cells, and those Schwann cells again can help form this kind of repair tube if the axon is damaged, okay? So again, that's kind of really all you need to know, is that in the peripheral nervous system, that can happen again, partially because again, we have that neurolemma that we don't have in the central nervous system, okay? So that's really important because remember, in the central nervous system, they have the oligodendrocytes that make the myelin sheath. So neurogenesis in the central nervous system, again, we really didn't think that new neurons could form from stem cells after birth, and especially, I shouldn't say after birth, but especially after adulthood. So we thought once you become an adult, all the neurons that you have are the neurons that you have. And so if you damage those neurons, say goodbye to them. They're gone. Now we found out that that's not true of all areas in the brain. So for example, in the hippocampus, we have found, of course, this is an area that's important for learning. So it makes sense that the hippocampus would be an area where you might have some neurogenesis. So you do get some, again, it's limited, but you do get some neurogenesis in the hippocampus, as well as they found in the olfactory regions of the brain. Okay, so those are kind of the two areas, but for the most part, there is really a lack of neurogenesis in other areas of the brain and spinal cord. And this is why, again, you have damage to the spinal cord and you lose that function and you can't get it back, which is a super big bummer, right? So that's why. Factors that prevent neurogenesis in the central nervous system. So we have, and this is a little bit more than you need to know, but inhibition of neuroglial cells, the absence of growth stimulating factors, lack of neurolemmas. Remember, we don't have neurolemmas because we don't have that, we don't have Schwann cells, we have oligodendrocytes instead. And then rapid formation of scar tissue is a big one, okay? And this becomes an issue for things like multiple sclerosis. So, knowing that rapid formation of scar tissue is important for understanding multiple sclerosis, which I'm going to talk about in just a minute. So let's talk about some of these clinical aspects. So impaired action potential impulse propagation, which just means being able, again, to propel that action potential all the way down the axon, can be caused by both chemical and physical factors. We already talked about some of the chemical factors that can block it, like local anesthetics that actually block the voltage-gated sodium channels. Things like lidocaine, right, can do that. But we didn't talk about things like temperature that can. So cold temperatures or continuous pressure, like I'm sure you guys have all done this. You've fallen asleep on your hand, right? And you kind of interrupted the blood circulation to your fingers. Then all of a sudden you wake up and you're like, oh, my hands, my fingers numb, right? It went to sleep. And that's because you just interrupted the blood circulation that was bringing oxygen to those neurons. And so that's why. Also, if you've ever had really cold fingers and they feel numb, again, that's why cold temperatures are just decreasing the blood circulation and the oxygen delivery to those neurons. And then it's also really cool, the same reason why you ice an injury, right? If you have a really painful injury, you're going to put ice on it for this exact reason. So kind of cool to know why ice actually works. There is an actual reason as to why ice helps decrease pain. So let's talk about multiple sclerosis. We're going to talk just about a few diseases and really basic. I like to talk about multiple sclerosis, which is MS, because I actually did clinical research with MS. So this is an autoimmune disorder, and it's an autoimmune disorder that unfortunately attacks. So it means your own immune system is going to actually attack the myelin sheaths in the central nervous system. So it's attacking the oligodendrocytes in the central nervous system. And so remember that in the central nervous system, we can't, once the axon is damaged, we can't repair that axon in the central nervous system. It's not like the peripheral nervous system. So what ends up happening is again, as that myelin degenerates, as the immune system attacks it, and it'll attack it in multiple areas. Eventually, then that those areas that were attacked will develop scar tissue or plaques, and we call those sclerosis. So, hence the name multiple sclerosis. Obviously, this slows down the impulse conduction because now, if you don't have the myelin sheath around those axons, remember that impulse conduction is slower. And so a lot of times patients that are having active attack, are not able to maybe if they've for motor function to a certain area, they are not able to move that area as well. Okay. Like, they have decreased ability to walk. And demyelinated axons oftentimes will increase their sodium channels, causing these like cycles of relapse and remission. So what ends up happening is, those demyelinated axons, initially we start to see, the impulse conduction slow until all of a sudden the, sodium channels production increases on those. And then all of a sudden our impulse conduction picks up. And that kind of gives us that gives you that sense of remission. And they actually call, there are different forms of it, but the most common type is relapse remitting. And this happens because of this process right here. It's about 500,000 people in the United States that end up with MS, and it primarily affects young adults, usually in between the ages of 20 and 40. Oftentimes, interestingly enough, most of my patients can actually pin down what they think their trigger was for their autoimmune disease. So it's oftentimes usually a really traumatic event, maybe a really awful car accident that really overstimulated their immune system, and then again stimulated this autoimmune disorder, this underlying autoimmune disorder that they had. And then oftentimes you see that it's more common in females. So just kind of some interesting facts on MS. Epilepsy is the second most common neurological disorder. It affects about 1% of the population. And as you can imagine, there's a lot of different causes of epilepsy. Sometimes it's brain damage. Sometimes it's metabolic disturbances, infections, toxins. Sometimes vascular disturbances. You have issues with vascularization in the brain, head injuries. I actually have a close friend of ours had a major head injury and has suffered from epilepsy ever since then. And then obviously brain tumors can also trigger this. And epilepsy ends up being like this kind of chaotic firing of neurons. Okay. That causes, again, the characteristic epileptic seizures that you see. All right, so that's it. Sorry, I tried to make this shorter, and I know it wasn't that short. But let me know if you have any questions, and we'll stop this.